RBCs or red blood cells are important in human body because they carry a pigment known as hemoglobin. This hemoglobin pigment binds with oxygen and supplies oxygen to the tissue when there is need of oxygen delivery. Now, 2,3-bisphosphoglycerate is an important, or 2,3-BPG is a very important molecule which is one of the key regulators of oxygen delivery by hemoglobin. Now, for long, this question plagued the biochemists and doctors that how this 2,3-bisphosphoglycerate comes within the RBC. To explain that, doctors came up with a solution or hypothesis known as the Rapoport Leubering Shunt. So what happens is that RBCs, as they carry oxygen, they do not have mitochondria. Why? Because if there is presence of mitochondria, then there will be presence of electron transport system, and that will lead to oxidative phosphorylation to produce ATP. And if you have oxidative phosphorylation going on, then oxygen will get consumed, and hence RBC will not be able to effectively transport oxygen if it starts using oxygen on its own. That's why there is no mitochondria within the RBC, and only source of ATP for RBCs is glycolysis. So RBCs take up glucose from the outside, and this glucose undergoes a pathway known as glycolysis to produce pyruvate and energy. So this pathway that we can see over here is known as the glycolytic pathway. But there's a small shunt or sideline pathway that occurs in RBC from glycolysis. What happens is that there's one intermediate in the glycolysis known as 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate undergoes a change into 2,3-bisphosphoglycerate. That means one phosphate molecule gets transferred from C1 to C2 in that molecule in presence of an enzyme that is known as 2,3-bisphosphoglycerate mutase. Mutase are a type of enzyme which transfer functional groups from one carbon to another carbon atom. And this is how 2,3-BPG is produced within the RBCs. And this 2,3-BPG modulates or regulates the oxygen delivery in the tissue when there is need. Now, when there is no need of high levels of BPG, or you need to decrease the levels of 2,3-bisphosphoglycerate within the RBC, then you have another enzyme that is 2,3-bisphosphoglycerate phosphatase, which converts it into 3-phosphoglycerate or 3-PG, which is an intermediate of glycolysis. And this is how this 2,3-BPG again goes back into the glycolytic pathway. So this entire thing starting from 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate and again back to 3-phosphoglycerate, this is known as the RL shunt or Rapoport Leubering Shunt.